Hi, Miss Nikki here. Welcome to Chapter 25. This is Part 2, where we're going to talk about gene expression, but more about how do we control gene expression. When we talk about control of gene expression, we have to think about the fact that we have lots of different cell types. They're all going to have DNA. They're all going to have the same DNA, but certain genes are going to be turned on and certain genes are going to be turned off. There's no point in wasting energy in having a gene on if it doesn't contribute to the function of that particular cell. So a great example here, if we have a red blood cell, we have some housekeeping genes, and we have hemoglobin. This is the protein that carries oxygen for us. We probably want to turn that on and have it on all the time, right? Or else our red blood cells wouldn't be able to carry oxygen. In muscle, we're going to have the myosin protein turned on. Myosin is the protein that allows your muscles to contract. In pancreatic cells, we're going to have the insulin gene turned on so that we can make lots of insulin protein so that our cells can take sugar in and use it to produce ATP. Here we have a system that bacteria use. Again, a lot of times when I think about control of gene expression, it's kind of the waste not, want not. Why make a particular protein if you don't need it? So here's a gene, and this gene codes for lactase. It's an enzyme that breaks down lactose. So if there's no lactose present, if there's no sugar present, does this bacterial cell need to make lactase enzyme? No. So we have a gene here that codes for messenger RNA, which then makes a protein, and that protein is a repressor. It goes over, it binds to this particular section in front of the gene, and it represses. It says, don't make this protein. We don't need it. Well, guess what? If lactose is present, the lactose binds the receptor, and it triggers that gene to make the protein lactase. So now this bacteria can use the lactase to break down the lactose and use that sugar to create energy. Obviously, in eukaryotes, gene expression is controlled at many different levels. It's more complex. So we have pre-transcriptional control. So this is before we turn DNA into RNA. So here at the DNA level, we can have pre-transcriptional control. We can have transcriptional control. During this process, we can limit the amount of messenger RNA that's created. We have post-transcriptional control. That has something to do with the processing. Remember when we take premature messenger RNA and turn it into mature RNA? So we can have a post-transcriptional control. We can have translational control. That's when we take the messenger RNA and we turn it into the protein. We could have a control mechanism set up there. Or post-translational. Remember we have that polypeptide and we have to fold that polypeptide into a fully functioning protein. So we have various levels at which we can control the expression of a particular gene or the protein product that that gene is producing. Nice image for us to walk through these levels of gene control. Here we have pre-transcriptional. The DNA is wound around these histones, which are proteins. And if the DNA is so tightly wound that the machinery of transcription doesn't have access, so the RNA pol or polymerase cannot bind the DNA because it's wrapped so tightly around this histone. For transcriptional control, we have other proteins which are called transcription factors. And transcription factors can bind the DNA and drive transcription. Basically, read the gene, make tons of messenger RNA. But sometimes these proteins get bound or inhibited, and so that would slow down transcription. So transcription factors can increase or decrease transcription. Third, here we have post-transcriptional control. So this is in the processing from that immature RNA, messenger RNA, to mature messenger RNA. So remember the introns here are going to be removed, but we can leave some of them in. Sometimes there's going to be alternative splicing. So we can make multiple messenger RNA transcripts from one gene. Here at translational control, when the mature messenger RNA leaves the nuclear pore, depending on the poly A tail, how long it is, will tell you how long this messenger RNA can stay in the cytoplasm and be read. So it's possible for one messenger RNA to make multiple proteins, but it depends on the translational control. And then we have post-translational control. This is where we're going to do the folding from the polypeptide to the mature functioning protein. Sometimes we're also going to add phosphate tags, so we can modify the function of the protein in this post-translational control. Now we get to move on and talk about gene mutations and cancer. So if you have a gene mutation, this is a permanent change in your DNA. We have two different types. There's germline mutation, and that occurs in the sex cells, and it will be passed off to your offspring. And then we have somatic, which means body mutations, and these are not passed on. They're just present in you. Some causes of mutations. Some are just spontaneous. They just arise for the natural process of life and copying cells and copying DNA. Some are induced. A lot of us would probably think of environmental factors like chemicals. Being exposed to certain chemicals could induce more mutations. We do have some errors in DNA, but it's pretty rare. So 1 error per billion nucleotides when we're copying or doing that semi-conservative DNA replication. So we do occasionally have mismatched pairs. They're usually scanned, found, and replaced with the correct nucleotide, but we do have some errors. Most of us probably think of mutagens when we think about the causes of mutation. Obviously, an environmental influence or factor, you can see the list here: radiation, ultraviolet, chemicals, pesticides, cigarette smoke. We do have repair enzymes that are constantly monitoring and repairing, but there are some mutations that still occur. There's also something called transposons. These are jumping genes. Kind of funny name. What they do is they're specific DNA sequences that can move between and within chromosomes. So sometimes they can alter a neighboring gene. Remember how we talked about promoters, right? Here's your promoter DNA sequence, and in front of the promoter is a gene that we want to read. Well, what happens if this jumping gene inserts itself in the promoter, and now the promoter no longer works? So we don't know where the gene starts. That's going to affect protein production. Now that we know more about gene expression and the process to create a protein, we can think about how does the DNA affect the protein product. So sometimes you're going to have a DNA error and there's no effect at all. Sometimes you could have an error in DNA and it's going to cause an incomplete protein. What if a stop codon shows up early in the process of translation? You're going to have a smaller incomplete protein. It can also cause changes in specific amino acids. So sometimes if your DNA has an error, that's going to lead to a completely different protein from being made, just from one letter being off. And that can produce abnormal proteins. And we'll look at sickle cell next. So sometimes a single point mutation can make a big difference. Here's the gene in the DNA that codes for the protein hemoglobin. Hemoglobin is in your red blood cells and it allows you to carry oxygen. So during transcription, we produce our messenger RNA sequence. The codons are read by the transfer RNA and we build our amino acid sequence or our polypeptide. In this case, this is a mutant hemoglobin. So you can see that there's been an error. There's been a point mutation in the DNA. This adenine is supposed to be a thymine. Changing this one DNA nitrogenous base results in a different messenger RNA codon, which results in a completely different amino acid being added to the chain. And you can see this point mutation in DNA translates to a abnormal protein. And you can see in sickle cell disease, the red blood cells are this sickled shape, so they're not very good at carrying oxygen. So what we just saw was a base substitution, right? And it changed the amino acid sequence. You can also have an addition or a deletion. So we have an extra added nucleotide, or we've deleted a nucleotide, and either way, it affects not just one amino acid in the sequence, it affects all amino acid sequences after. Remember that messenger RNA reads in codons. So if you insert an extra, we call this a frame shift mutation, because now we have shifted the frame for every amino acid after the insertion or the deletion. So what happens if we have a non-functional protein? They want you to realize this can have a dramatic effect on the phenotype. So PKU, phenylketonuria, if you do not have this enzyme, remember a lot of what we code for in our DNA, those proteins are enzymes that are going to do jobs within the body. So if you do not have this enzyme, you're not able to break down PKU into tyrosine, and you have a buildup of phenylalanine, and it'll cause mental impairment. In another example, if you have this enzyme, you cannot break tyrosine down into melanin, and melanin is what gives you the pigment or color of your skin. So if you do not have the enzyme that allows you to break down the starting material, you don't produce melanin, and therefore you don't have color, you have albinism. Another example of what can happen with non-functioning proteins is androgen insensitivity. So your cells do not make the protein to recognize androgens. So here's your cell, and cells usually have receptors, and those receptors allow for items to bind, and it'll cause action in the cell, right? So what we're saying is certain people, they don't have this receptor. It's a protein. There was some sort of frame shift or point mutation. You do not make this receptor. So you have all of these cells that have no receptors. There is testosterone. You're a male, you're producing testosterone, but the testosterone just goes right by the cells. The cells can't see the testosterone because they don't have the receptors for it. As a consequence, these males are going to develop female genitalia. They're going to develop secondary female characteristics. So genetically, if we were to look at the DNA, the karyotype, they would be a male genetically, but phenotypically, they're going to look like a female. So how can these mutations cause cancer? Imagine if you have a tumor suppressor gene. Obviously, it suppresses tumor development. What happens if this gene becomes inactive? You have a mutation, you're no longer making the proteins that are going to stop tumors from developing. I do want you to realize that these tumor suppressor genes are going to code for proteins, and those proteins are going to stop tumor development. I want you to realize that they can be inherited or acquired. So inherited, let's say you're supposed to get 2 good copies. What happens if one of your copies is defective? Now you only have 1 good copy. Okay, now acquired, the environmental factors. You smoke, right? You work with pesticides, in a chemical plant, you're out in the sun a lot. So you can see that there can be a compounded effect. It's not only inherited but also acquired that could lead those mutations to cause cancer in an individual. One of the most common tumor suppressor genes is called P53. P53 acts as a transcription factor and it controls the cell cycle. So if you're missing P53, your cell cycle continues to copy, right? Copy cells, even if they're defective. And it also promotes apoptosis. So apoptosis is cell death. If you have any sort of DNA mutation, apoptosis can be triggered during the cell cycle. So you can see if you're missing P53, your cell cycle just keeps copying and you never catch the mistakes and tell the cell to do programmed cell death so that you don't keep copying those mistakes. So not all abnormal cell growth is going to be cancer. You can have a benign tumor, or you could have a malignant tumor. So benign, it doesn't really grow, it's not cancerous. If it's a malignant tumor, it has the ability to spread throughout your body. So think of organs you would not want to have cancer, right? Pancreas, liver, gallbladder, intestines, right? Here we can look at the progression of cancer visually. We can see that this one cell has a mutation that's arisen. Through the copying process in the cell cycle, we now have developed a tumor. So this is not cancerous. You can think of it as precancerous. It is a tumor. But if the tumor starts invading your lymphatic system or gets into your blood vessel, if it can set up shop some distance away from the primary tumor, this is a malignant tumor. This is metastasis. Characteristics of cancer cells, things that we've noticed about them, is that they're genetically unstable. Remember at the beginning of this chapter we talked about one of the inherent properties of DNA is that it is stable, that we can transfer DNA from generation to generation with very little change. Well, cancer cells are kind of a mess. So one cell can acquire a mutation, and then eventually those are going to continue to divide. We can have more mutations, multiple mutations, and those multiple mutations can lead to chromosomal rearrangements. The second characteristic is that cancer cells do not regulate the cell cycle correctly. If you remember, the cell cycle has stopping points, kind of checkpoints, and if something is wrong with the cell, you'll see apoptosis occur, cell death. So these cancer cells are going to increase the rate of cell division and increase the number of cells. Third characteristic, cancer cells can escape the signals for cell death. So usually if there's some sort of genetic damage, we're going to signal apoptosis, cellular death. Cancer cells do not respond to that internal signal to die. Also, we have telomeres, and the length of telomeres are kind of like biological clocks for cells. Cancer cells do not have a limited number of cell divisions. They basically avoid the telomere time clock and they just keep dividing. So this is, sometimes you'll hear cancer cells listed as immortal. They'll just keep copying themselves and they aren't listening to the signals to die or, hey, you've divided too much, your time is gone. And the fourth characteristic, cancer cells can survive and proliferate elsewhere in the body. So we already looked at this term metastasis, basically when these cancer cells are traveling through the blood or lymphatic vessels to new tissues and new organs and forming new tumors in those areas. Another interesting fact is that cancer cells can trigger angiogenesis, or the creation of blood vessels that are going to feed the tumor, give it blood and nutrients and oxygen, and basically deprive it from the normal surrounding tissues. So the tumor is basically invading and stealing all of the resources, and it's outgrowing the normal tissue. Okay, well, that's it for chapter 25. Thanks, guys. I'll talk to you later.